Thank you for coming. So today I'm going to be talking about shedding light on inconclusive DNA and TrueAllele computer analysis. And to start off, as an example, I'll be talking about the beer bottle in this Ronald Meadow case that Bill Fitzpatrick litigated. And what I'm going to go through are the slides that we prepared for the jury trial, which will give you a sense of how this gets presented. Though the Meadows case, the defense stipulated to the TrueAllele evidence, and so this was not shown at trial. So, just some background. DNA biology. In the human body, there are trillions of cells, and most of them have nuclei. The nuclei have 23 pairs of chromosomes. Each chromosome, if you unravel it, is just text, like on a file or a book or a hard drive of a computer. It's just text that gets read. It's a string of letters written in a four-letter DNA alphabet, A, C, G, and T. And the markers of interest in forensic science are markers that can distinguish between people. And so there are short tandem repeats. I'll explain what they are. They were identified by geneticists 30 years ago for the purpose of distinguishing people from each other because they have a lot of variation. So if you think of the human genome, this encyclopedia of 3 billion DNA letters, we have two complete copies, one from each parent, in each of these trillions of nucleated cells. And if you open up to a paragraph that has a short tandem repeat, this is what the paragraph looks like. It has a starting phrase, take me out to the ball game, some stuff in between, and then an ending phrase, at the old ball game. This is a famous American poem, right? Take me out to the ball game, take me out with the crowd, and so on. And then in the middle, there's a word, usually 4 or 5 letters, that's repeated some number of times. In this example, it's repeated 10 times. It could be 5, 7, 13, 21, 28, whatever the number of repeats is. And everything else is the same about the paragraph. But that's the only variation, because the variation of the number of words changes the length of the paragraph. If the word root is repeated more times, then the paragraph is longer. So scientists can find that paragraph chemically by putting in the starting phrase and the ending phrase. And that's like a string search that you would do in Google or a legal search, and you'd find that paragraph chemically anywhere in the encyclopedia. And they can amplify this paragraph millions of times and label it. And by measuring the length of the paragraph, they can figure out how many times that word root is repeated. And since it's repeated usually differently in different people, that's a way of distinguishing people. So that's the source of variation. You don't need to know what volume it's in. Just the beginning and the end string search finds it. And a scientist will do this for a multiplex in a tube of maybe 10 to 20 different paragraphs simultaneously. That's the source of DNA data. And an allele is the variation. It's the number of times the word is repeated. So here the allele is 10. The key concept in forensic DNA is the genotype. That's what you're trying to measure and find in different people. If you open up a chromosome and look at a location and look at the alleles, which scientists do chemically with these STRs, you'll find that there are 2 different alleles, one from the mother, one of the father. They can have the same length or different lengths, depending on whatever the person inherited from their parents. In this case, that middle section of the paragraph is 10, because the word, some 4-letter DNA word in ACGT, alphabet is repeated 10 times. And say from the father, randomly from the population, that word is repeated 12 times. And so, therefore, the 2 alleles are a 10 and a 12 at this one location on a chromosome in that genetic encyclopedia. And so the genotype, the genetic type at this location of that individual is the pair, 10, 12. And because there are many different alleles for a location in the population, maybe 15 or 20 different alleles, and we're looking at pairs of alleles, not just one allele, there's about 100 possible genotypes at each of these locations that scientists look at. So if you multiply all those possibilities together, 100 times 100 times 100, 15 times, that's where you get like a septillion possible different genotypes. There are lots more genotypes than there are people, That's why they're good for distinguishing people. This is how DNA is interpreted on easy DNA and the basis of the paradigm from maybe 15 or 20 years ago, when DNA was simple and abundant and it was just easy. Again, this is an historical relic from the 20th century. But you'd have DNA from one person. You have a lot of it. The laboratory would extract the DNA, amplify it, separate it out by size at maybe 10 or 15 different locations. We're just looking at one location, one locus. And there'd be a signal. And the signal going from left to right is based on size. If you measure the length of the paragraph. And then by calibrating it, scientists could say, oh, that size corresponds to 10 repeats, an allele of 10, and one that's longer may have 12 repeats. And so the 10 and 12 in the human genome would turn into a data signal that would have two peaks, one at 10, one at 12, the number of repeats. And just by looking at that, you could infer and say, well, the evidence genotype must be a 10-12. It's just evident from the data. That's how DNA used to be that easy most of the time. After that's been inferred, then a comparison can be made. And if the person actually has a 10-12, you can make a statement about match or inclusion. But nowadays, most DNA is mixtures. In fact, it probably already has been. It's just we're looking at more interesting DNA, and the machinery and the chemistry is more sensitive. And these mixtures may have 2, 3, 4, 5, 6 people in them. If you take a look at a lot of casework, you'll see that. Here's an example. Suppose there's 2 people in the mixture. The lab does the identical procedure of amplifying the DNA and separating it out by size, but now instead of there just being the 2 alleles from one person's genotype, there's more. And so maybe there's a pattern of 3 alleles. Now the peaks have more than 2 alleles. They also have a pattern. There's different heights. Some peaks are lower, some peaks are higher. And the height of the peak is not identical to, but it corresponds to the quantity of DNA. Typically, taller peaks will correspond to more of that allele present in the test tube at that location. And we're just looking at one location. Now, when an inference is made, the lab can say they can't, well, maybe they won't reach a conclusion, or it's too low. But if they do reach a conclusion, the way to interpret this evidence is to separate out the genotypes of the different individuals. So let's look at, say, one of the individuals that contains the 11 allele. There are maybe 100 possible genotypes before you saw the data, a 5 and a 5, a 5 and a 6, all the way up to maybe a 20 and a 20, all those possible allele pairs. What the data does is it concentrates the answer to just those possibilities that are more probable. And you can still interpret it, but now instead of saying it must be a 10 and 11 or it must be an 11-11, there's a list of possibilities, maybe a shorter list of possibilities, and they get probabilities. I'll show you how that happens later on. And the probabilities are just numbers, either fractions or percentages that add up to 100%. And if you have a good way of objectively inferring these genotypes for each of the contributors, again, this is for one contributor that contains the 11. There's the other contributor that we saw before that was the 10 and the 12. This separates out the genotypes from the mixture. That's really the key idea in modern evidence interpretation for mixtures, to unmix the mixture and separate it back out into the genotypes of the different people who contributed their DNA. Once it's been separated out, then it's easy to make a comparison with a known genotype and determine the extent of match. Okay, so in essence, what I'll be talking about today is unmixing mixtures, separating them back out. Now, computers can use all the data. In a case like this, the lab didn't use the data. I'll be talking about why later on. It has to do with the quantity of DNA and the peak heights, but most mixtures in the US are not interpreted, or they're not given a meaningful match statistic. But computers that know how to can use all the data. And here we're looking at the quantitative peak heights from one of the loci, has a name, O1. The fact that it's called 9.3 instead of 10 has to do with the chemistry, but it's still length that's more than 9. And on the horizontal x-axis, this is the size of the paragraph, the number of letters in the paragraph, like 172. The number on top is the number of repeats of the four-letter word in the THO1 locus. And we see that some peaks are taller than others. 6 has a height around 100, whereas 9.3 has a height of around 50. In addition to having different peak heights, you also see that there's a pattern. There's a pattern that has to be explained of why is it that we have those 3 different alleles with tall peaks present in that configuration with some high and some low. That's a lot of information that a computer through statistical modeling can address. Okay, so computers use all the data. How? Well, with modern computing, which started after World War II, computers have a lot of brute force strength, but they also can build up mathematical and statistical models to solve probability equations. And the way they do it is they try out, by statistical sampling, many, many possibilities. Thousands, or in a case like this, tens of thousands, sometimes we do hundreds of thousands of different explanations at each of these genetic locations. And this is what an explanation looks like. Suppose that there's a major contributor. The computer is proposing this. That's a 6 and a 9, shown as blue rectangles. It's a pair of alleles. And the heights represent the amount. And there's a lesser amount of a person whose genotype is a 9.3 and a 9.3. They have 2 of the same allele. Then a pattern that proposed that one person contributed a lot of a 6-9 genotype, and a second person contributed a lesser amount of a 9.3 genotype, would produce a pattern in these orange and blue rectangles that explains the peak heights of the green signal underneath. That is, the heights of the rectangles, when you add them all up together, are close to the tops of the peaks. That's a good explanation, and that means that the 2 pictures look the same. The 2 patterns look the same, of the underlying data in green and of the genotype explanation in color. Now, what the computer does is it moves those rectangles all around, trying out lots and lots of different genotypes. It may try out a genotype where it tries out a 7 and a 10. There's no data there, so it has lower probability. It just doesn't explain the data as well. But in order to follow the rules of probability, it has to try out lots of possibilities where the data aren't as well as where the data are. Those will have probabilities near 0. But it has to try out everything to be complete. When it's done trying out all of these possibilities, it arrives at, for this locus, for one of the contributors, what the genotype is, which is going to be, as we saw, a listing of different possible values of allele pairs with probabilities attached. And it does this for each of the contributors at every locus. So at the THO1 locus, for one of the contributors, this is what a genotype looks like. 15 years ago, people may have tried to say, well, it's only one of these possibilities, or they may have said nothing. Nowadays, you can say, here's the list out of 100 possible values, 66, 69, 99, and so on. Here are the 4 or 5 where most of the probability is concentrated after trying out all those different possibilities. Okay, the computer's doing this for all the different genotype values at once, as well as dozens of other variables that are really of interest only to scientific specialists, stutter and stuff. Okay, so this is a genotype. At this THO1 locus, from this beer bottle, the computer has separated out the mixture into 2 genotypes. This is one of them. And this has been done objectively just from the data. The computer has not seen Ron Meadow's genotype yet. It hasn't seen anybody's genotype yet. It's only seen the evidence. All this is done to get the genotypes out of the evidence, because once they're unmixed, it's easy to compute match statistics. And this is how you compute a match statistic. This is the most technical slide, except for the whole bit about how the computer works. So DNA match information. The DNA match statistic is answering the question of how much more does an individual, a suspect or person of interest or victim, match evidence than a random person. And this is how it's done. We start off with a separated genotype of one of the contributors at a locus. That's in blue. And then in gray or brown underneath, these lower bars correspond to what is the chance that a random person in the population or in this room would have a particular genotype. Again, there are hundreds of possible values. I'm only showing the half dozen that are relevant to making the comparison with the evidence. So the evidence is in blue. That's what the computer separated. And the population, the random person, is shown next to it in brown. And then the question is, what is the difference? Now, to do that, now for the first time, you have to pick somebody you want to compare with. And it turns out that Ron Meadow's genotype at BO1 happens to be a 6'9". The computer didn't know that until now, but now it'll make the comparison. You compare it with anybody's genotype. So we now focus inside this red rectangle at 6'9", and the math of match statistics lets us largely ignore everything else. We ask, what's the ratio of the separated evidence genotype, the blue bar, at 58%, divided by a random person having that genotype at 5% in brown. And that ratio of 50 to 5 is around 10. That's the match statistic at this one locus. That's what a match statistic is. But now, in addition to having that one location, the computer has separated out the genotypes at all the locations. The comparison can be made to a suspect or a person. And these match statistics, shown on a scale of 10, 20, 30 horizontally for these 15 loci, are calculated and multiplied together to produce a number, and that number gets turned into a match statistic that's written into a report. And so, focusing on the Caucasian population, we can numerically answer the question, to what extent is the suspect in the evidence? And as we wrote in the report, a match between the beer bottle and Ronald Meadow is 28 million times more probable than coincidence for a Caucasian population. So that's what the match statement looks like. The reason you can make a match statement this simply is that you're actually comparing two probabilities, the probability of a specific evidence match to that of a coincidence, and also because the computer separated out the genotypes. So now you have the simple language that's plain English of probabilities of matches, because it's back to comparing a single genotype contributor from an unmixed mixture to a person. Now I'm going to be moving into something else in a few minutes, so I want to introduce a concept for you. When scientists measure information, numbers like million and septillion and 1 in a quadrillion are they're very big numbers and very obscure words. Information theory has us look at just the number of zeros. So here, 10 million is a 1 followed by 7 zeros. 100 million would be a 1 followed by 8 zeros. So this number has 7 point something zeros in it. That's called a logarithm. I'm not going to say that again. It's just the number of zeros in the match statistic. And here, that number is 7 or 7 point something. And that's the measure of information. You'll see immediately why it's good to have a scale that can go from 30 zeros to no zeros, which is 1, to an exclusionary statistic like 1 over or 1 followed by 30 zeros, and a scale where you're just counting the number of zeros. And so this was something else that was in the report, and it's an important concept for understanding the reliability of methods. Suppose we were to compare the evidence genotype that we obtained from the beer bottle against someone who didn't contribute their DNA to the evidence. We can calculate match statistics for that. There are a number of ways of doing it, but we do that in our system. And so the bell curve that you get is the distribution of the non-contributors to the evidence. That's what you would see. And here, the number is maybe around -6 or -7. That's 1 over a million. So that's the exclusionary power if somebody did not contribute their DNA. And compared to that bell curve, that's Ron Meadow. He's up at 7 zeros, not down at -7 zeros. That's a nice graphic for just showing what's the chance we're wrong. As you know, in the end, Ron Meadow was found guilty of murdering his wife. So I'd now like to move a little south to Virginia and tell you about a story in Virginia and the studies that came out of it and really what happened across the country. This was an interesting headline on the Washington Post 3.5 years ago. Virginia reevaluates DNA evidence 375 cases. What had happened, and I'll be telling you more about the history of this, is that there was a rules change in interpreting DNA evidence. And when the rules changed, the lab recalled 375 cases and said that the match report that we sent out to our DAs, we have to take back because we can't support those match statistics with the new guidelines that came down from Washington. So let me just think about, what does that even mean, that the rules changed and suddenly the report that you're holding on to is not a report anymore? So what Virginia did is they asked the question, can we do this within the guidelines using a computer? Because manual methods of review, as various famous people talked about, have a lot of problems with mixtures. And so that's how we got involved with Virginia, and we started preparing reports for them. So let me tell you what they were doing before 2010. And this is what most labs still do. It's part of what labs still do for the human review, whether they're doing it manually or using a computer. Instead of looking at a pattern of 2 tall peaks from a major contributor and 2 shorter peaks from a minor contributor and trying to separate out a genotype, what they do is they draw a line. And that line is determined in a variety of different ways, supposed to be indicating separation from background noise, but however they determine it, it's usually a number between 50 and 200. They draw this line, shown in red, and any peak that's above the line is in, any peak that's under the line is out. There's no real theory behind this. It's just a procedure that came from the FBI about 20 years ago that people follow. It's in a lot of their software for computing statistics. And after they draw the line, those 4 alleles in this example and all the different pairs that they have of possible values are now part of an inclusionary result. But the problem here is that if the correct answer is really obviously just a 10 and a 12, all that probability is spread out over all these different answers, and the power of the match statistic decreases. How much? Well, we'll start making comparisons, but on average, now this is the same scale that you looked at before, where this is the number of zeros in the match statistic. A forensic scientist call it the logarithm of the likelihood ratio, or the log LR. But it's just the number of zeros in the match statistic. 0 is a 1, 9 is a billion, 18 is a billion billion. I guess 27 would be a billion billion billion, right? But this is just the number of zeros, and this is the amount of information. You see it's pretty concentrated, and it's between 6 and 7 zeros. The average is 6.5 or so. And the average match statistic is between 1 million and 10 million. That's the information that labs used to produce before 2010 by computing a combined probability of inclusion, or a CPI statistic that you've probably seen in some reports. That's the old CPI statistic. They just add up the frequency of the alleles that are over a threshold, square them, and then that's the number. It's a very simple procedure. And that's how much information it used to produce. These data that you're seeing are taken from the study that we did with Virginia, looking at 100 match comparisons and how much information was in those comparisons. And part of the study was 80 human comparisons that Virginia looked at. That's where this data is coming from. This is what their match statistics would have been. But there were a lot of problems with threshold methods, and organizations started doing studies. The National Institute of Standards and Technology, or NIST, which I'll be mentioning a few times, has an applied genetics group. Dr. John Butler of that group presented in his 2005 study on mixtures called Mix-05 some results at a number of conferences. And this is the key slide that gets shown over and over again. And it's mentioned in the New Scientist and other articles. And that is, on the same 2-person mixture, the range of information of the 50 or so labs given the same data that gave an answer. Many of the labs said it was inconclusive, but the labs that gave an answer gave numbers ranging from tens of thousands to hundreds of trillions. that is information from 4 zeros to 14 zeros on the same mixture. Okay, and so basically the threshold methods were shown to not be very reproducible. You might also ask, if the real number was actually more like 15 zeros, then what accuracy do we have in human mixture interpretation with thresholds? Why are you getting an inconclusive or a 31,000 and everything in between if the real answer is a quadrillion? So this raised a lot of concern within the forensic science community, but not so much outside. Probably because you didn't hear about it. And so the Scientific Working Group on DNA Analysis Methods, which is run under the auspices of the FBI for setting guidelines for how labs should interpret DNA, said, well, maybe we could add a second threshold and have a higher threshold, and that would provide more consistency. Maybe if we did that, we'd say that you can't report a match statistic unless all of your peaks at the lower threshold also reach the higher threshold. In this case, you throw the baby out with the bathwater, that information would just be lost. And maybe that would be a more conservative way of doing things that would solve the problem, and we get rid of all this inconsistency. So that's just what was implemented. And you may have noticed that around 2010, your match statistics got lower, or there were more inconclusives. And that was a national trend. It wasn't, just your lab. Everybody did this across the US. And this is why. And then the amount of information, if you're throwing out a lot of your data, and you say we'll still report an inclusion, but with a lower match statistic, we saw a shrinkage to the left. So in this study, Virginia had scored about 50 of these 100 reported matches that we did by computer later on, using this stochastic threshold, or modified CPI level with a higher threshold, and the average match statistic dropped to 140, which is 2 zeros. So you see a reduction of going from more conclusions to fewer conclusions, and an average match statistic shifting to the left from around 6 to around 2 zeros after the 1. This will happen around the country, but Virginia documented it. Did this solve the problem? No. NIST did another study with 100 laboratories sending in results with different mixtures. This is a 3-person mixture. This is the Mix 2013 study, or Mix 13. This is an interesting slide that NIST has been presenting at talks around the country. So these numbers add up to 100. There were 100 labs that replied. And 70 of the labs reported an inclusion to a contributor who was not in a mixture. So 70 labs gave a statistic ranging from 9 to 344,000 in this study to somebody who wasn't in there. Another 25 labs said we couldn't say anything, and 6 out of the 100 labs gave an exclusion to the suspect who was not present in the mixture for various reasons, and one of them had a negative number of zeros, a negative match statistic, and that group was using TrueAllele. I'm going to tell you more about TrueAllele. This is what TrueAllele looks like. If you had it in your office, you'd have this big screen computer, a Windows or a Mac machine. In some back office, hopefully, not on your desk, there'd be these other computers. There's a database. There's other parallel computing. Some groups can have up to 60 or 80 processors. Most groups just have 4. And the data is entered, questions are asked, it's all uploaded to a database. The computers go off and think about it for hours, usually hours. If it's a very hard problem, maybe days. The answers come back, they're written, and then a user can look at the answer in a very visual way, looking at mixture weights and genotypes and match statistics and data to review a case and prepare a report. The system took 10 years to build, going through 25 versions, because it worked right out of the box in 1999, but it didn't always give the right answer. And at Cybergenetics, we were very focused on always giving the right answer. And that meant making computer models that put in more and more variables. Instead of having 1 or 2 variables, the system may consider 100 different variables and all their uncertainties as it's solving a problem. So it's a very rich statistical model, and we were testing it continuously. And then after that was done, we then spent another 5 years extensively testing it and validating it, usually with other groups. And so these are 7 peer-reviewed validation studies that talk about the reliability of TrueAllele. The top 4 are done on samples of known composition synthesized in a lab. The first was published in 2009, describing an information gap in the sensitivity of human review and computer review. Human review methods went down to about 100 or 200 picograms of DNA, and then they stopped. The computer could go down tenfold more, to 10 or 20. This is a paper written with Jack Valentine, who is actually chair of the DNA subcommittee in New York, about DNA mixture genotyping using probabilistic computer methods. It talks about low template DNA that people couldn't resolve. I should say that these and most everything else are on our website. You can actually download whatever available. If there's publication restrictions from copyright, if you email me, I can send you anything you'd like as a correspondence. This paper is done with a California lab with Dr. Kevin Miller, and this explores what happens when you go out to 5-person mixtures, and you say that there's 5 unknowns we have to separate. So it looks at 2, 3, 4, and 5, low amounts of DNA. And this is an independent study by Virginia, Dr. Susan Greenspoon, that'll be appearing next year, establishing the limits of TrueAllele. The next 3 papers are based on casework, actual cases. This was done in collaboration with Dr. Barry Duesman at the New York State Police Forensic Investigation Center in Albany. And it was work that was done as we were preparing for the DNA subcommittee to get approval for the Albany lab. And it's entitled Validating TrueAllele DNA Mixture Interpretation. It was published in the Journal of Forensic Sciences in 2011. This is a second paper with Dr. Barry Duesman, the New York State TrueAllele Casework Validation Study. I'll be showing you some quick results from this later on. It's a much more expanded study that was also presented to the DNA subcommittee. This paper was done with Virginia, and that's the basis of what I'm showing you now with all these comparisons. The main collaborator was Dr. Susan Greenspoon. It's published in an online journal that's freely available. So if you just type in a few keywords from the title, it's there. It's actually had over 2,500 views, which for a highly technical paper about DNA mixture interpretation is about as greatest hit as you're going to get in this field. And that is TrueAllele Casework on Virginia DNA mixture evidence, computer and manual interpretation in 72 reported criminal cases. Because the conceit of this is we did the work on 72 cases as an extension of the lab before they brought it on board themselves. And within those 72 cases, there were 100 match comparisons that we're able to report on 2, 3, and 4 contributor mixtures. So I'm going to tell you some of the key results from this study. And the main 3 words are sensitivity, I'm going to show them and explain them, are sensitivity, specificity, and reproducibility. And what does that mean? Sensitivity is the ability to detect something in science. So in this case, it's the extent to which you correctly identify that there's a contributor is in the DNA. That if somebody's in there, you're finding them, and they're true inclusions. And out of 100 reported matches, there are 80 with a statistic over 1 million, and here's the distribution. So ranging from a match statistic in the hundreds, that's 2 zeros, up to a match statistic that's close to 24 zeros. 24 divided by 2 is 12, and 1 followed by 12 zeros is a trillion. So that's a trillion trillion. That's why we use numbers. It's hard to remember all these words. But what we get is a full range of match statistics from something that's low in the hundreds to something that's higher, that's more like a trillion trillion, and all the numbers in between. And the average here is 11 zeros, because it's the full spectrum. And that corresponds to a number in the hundreds of billions. So this is the same data that can be looked at by thresholds, pre-2010, stochastic thresholds, post-2010, or by computer. So this is what we reported on in that study. Now, you may say, well, isn't it just that it's software? No, there's a lot of software in there. There's a lot of new software in there. But the goal of most software is to produce a number that a lab can use. It doesn't have to preserve all the information or give the most accurate result. That's what cybergenetics was hung up on for 15 years. Most software doesn't do that, and labs are not evaluated on the numbers they produce or their accuracy, or even their ability to produce a number if they're going to report a result. So here's a comparison that was presented this summer at a conference on forensic inference and statistics. This is not my result. If you look at the blue curve on top, what the x-axis is showing is what happens when you have a lot of DNA, a nanogram, less DNA, half of that, and then even less DNA, a quarter of that, and you're starting to get into low template territory. And scientists sometimes look at the number of alleles they know that are present, but they can't see if they're doing a study where they know the answer. So if the peak is under the threshold or not there at all, and they count the number of dropout events, 2, 8, and 14. What TrueAllele does, this again is the number of zeros in the match statistic, log LR, is it starts off around 13, and then as the amount of DNA falls, it goes down to 9, and then down to 7. So 7 is 10 million, a 1 followed by 7 zeros. That's consistent with the studies that we've published. That's what TrueAllele does. As there's less DNA from a contributor, you get less information. And these other programs, StarMix, LRMix, LabRetriever, they have different methods. They all have dropout parameters and thresholds, which TrueAllele doesn't use. And on easy cases, they start off either using more of the data or more like a threshold method. That's why you're seeing the 2 lower peaks around 6. But as the amount of DNA dropped in this study, the amount of information disappeared as well. So it's not just that it's software, it's that it's evolved software that's been extensively tested. Next, specificity. Well, you don't want to include somebody who's not a contributor in your DNA. You don't want to falsely include and ultimately falsely convict, imprison, and execute, or however far it goes, based on wrong DNA evidence. So this is the extent to which the interpretation does not misidentify the wrong person. So we want to measure true exclusions without false inclusions. The way we did this is we compared our 100 inferred genotypes that we reported on and compared them to tens of thousands of randomly generated genotypes that were not included. Right? Because there's so many more genotypes than people. If you generate random genotypes, they're not included in particular mixtures. And so then we were able to look at the distribution of the negative match statistics. There's a lot more in the paper where we look at the exact number of false positives. So I didn't want to bore you with all that today. Judges hear about all this at admissibility hearings. So now what do we have? 0 is now over here. So this is no information, a match statistic of 1. Here is -9, which is 1 over a billion, which is an exclusionary statistic. And -18 is 1 over a billion billion. So on average, the exclusionary power, the colors are different ethnic groups, and this distribution is just telling you how much you get of each exclusionary value. It's just a frequency distribution. So the average is -19. Again, -18 is 1 over a billion billion. The inclusion methods that the labs use are not good for exclusion. I don't think you usually see exclusionary match statistics, like a person is not in here, or the chance of a coincidence is a billion times more probable than an actual match. But TrueAllele and its likelihood ratio statistics can do that. And this is the mismatch distribution. We showed that the system was very, very specific. We weren't falsely including people. The last axis is reproducible. For reproducibility, the question is, if you run the computer twice, do you get the same answer? Now you could ask, well, it's a computer program. Wouldn't you get the same answer? Well, yes and no. Modern computing, which again started with the advent of computing during World War II, the first algorithms were random algorithms for high-dimensional physics problems. If you only have a few dimensions, you can get an exact answer. But once you reach the point where you have dozens or hundreds of dimensions and you're solving probability equations, the computer solves these with statistical sampling. You sample from the answer. A way of thinking of it is that if you have a camera with a CCD and you expose it for like a millionth of a second, you don't get that much light. But the longer you let light hit it, the more signal you get. And it'll be different every time you expose it, but you get the correct picture if you expose it long enough. They're random algorithms, and that's standard in thousands of fields. It's just what people do for hard problems. So what we did is we ran it twice, and we looked at what the difference was. The first number is on the x-axis. The second number is on the vertical y-axis. If you pick some number here, like that point, you see that it has around a 10 on the first computer run and around a 10 on the second computer run. For comparison, if you were to think about what happened in that MIXO5 study with human review, if one person got a 4 zeros and the other person got 14 zeros, your points are lying out here. But here, but in this picture, in this study, what you see is the points are all lying along 45 degrees, or x equals y. So they're basically the same number with a little bit of variation. And that's what you get with random algorithms, and it's a lot better than human review by many orders of magnitude. Now here's a comparison of the methods. This is the old method, CPI, giving about 6 zeros. This is the stochastic thresholds method with 2 zeros. And this is TrueAllele, giving an average of 11 zeros. So you see the difference in information and in how much is preserved. This is 100 matches, 50 matches, and 80 matches. But now there's another concept, and that is this accurate? It's sensitive, it's specific, it's reproducible. How do you measure accuracy? Well, you can't really determine what the accuracy is of one particular match. But what you can do is say, if I have a large number of matches, an ensemble of 100 matches, how are they distributed? And you can answer it statistically, which we did in this journal article. If you have random mixture weights that are evenly distributed between 10% up to 90% with random amounts of DNA, what we expect is the number of zeros in the match statistic, the amount of information, will also be evenly distributed. As scientists, that's what we expect. And with TrueAllele, that's what we see. The dashed line corresponds to an even distribution, a uniform distribution of match statistics. And you don't see that, right? With red, with modified CPI, everything's to the left. There's nothing to the right. It's not uniformly distributed. The same thing with the older CPI method. There's more around 6, but nothing further to the right. And statistically, in the paper, we showed that there was no statistical distinction between TrueAllele's ensemble of match results and a flat distribution, whereas there was a deviation for the other methods. So this gave statistical support for accuracy by looking at a large ensemble of results. What were the outcomes in Virginia? Well, these are cases where the DNA had been withdrawn. This is about a year ago. There are more cases that have gone to court. But over 10 trials, these cases in federal, Marines, state court for various violent crimes resulted in, these were trials, resulted in convictions, and there were a lot more plea bargains than there were trials. So these were cases that had been brought back to life by computer interpretation. Okay. I'm now going to, that's the Virginia story. But now we're in New York, right? I flew up to Syracuse, not down to Richmond last night. So I'm going to tell you about New York State. In Albany, the crime lab has brought in TrueAllele technology. The first technology was a simpler system, the TrueAllele databank system that was for reference samples for the convicted offender database. And the New York State lab brought it online. They had a long period of time of validating it, getting FBI approval, which took longer than everything else put together. When it was all done, this is a slide from Dr. Barry Duesman, the retired, I think actually this month, as director. He'd been there for 25 years. And what you see, we're going through 2006, and their DNA backlog is increasing. They introduced the TrueAllele reference sample system, and their DNA is eliminated. That's what you'd expect if you bring in computers. Okay? And they check for errors, they don't find any. But what used to be a large operation was reduced to just a few computers. We also were involved in reanalyzing the World Trade Center data from New York City, looking at over 18,000 victim remains and 2,700 missing people with mixture data, kinship data, and then making comparisons and sending those results to the medical examiner. But getting back to TrueAllele casework and mixtures, Albany started going to the DNA subcommittee 2009, 2010, and began presenting studies. And each of these studies was turned into a paper. This is the first paper in one of the first studies. The question was, if New York State said that there was a match statistic, did the computer agree? That was the first question. And so in orange, we looked at 16 different mixture items in this paper. Here are 8 that use the CPI method. And in orange, you're seeing the results of the CPI match statistic. They're all around 7 zeros, around 10 million. And that's the result. Because you remember from the other pictures, CPI tends to always give the same number. It's always around 1 million or 10 million. That's what you get. The TrueAllele computer looked at the same mixture data, 2-person mixture, not assuming anything about other known contributors. And in blue, the bars are showing what it found. And on average, it found 6 zeros more, about a million-fold improvement of information wherever New York had called a result. So we then continued. Now we asked another question. It preserves more information, but how much more match information? This is a different study. Now we're looking at 81 mixture items that produced about 85 match results, because you can have more than one person in a mixture that's of interest. And now we're saying just on this population of data where there's a result, what did people do? Going the other way. This blue cloud, starting with number over 20 zeros in the match statistic and continuing down to the right for these 85 matches, is what the computer found. So those are its match statistics. On average, it's finding 15 zeros, about match statistic of a quadrillion, on this study with New York. And the different colored bars are different methods of human review. Gray is when you treat the mixture like there's only one person in there. Green is when you have a 2-person mixture and you assume the victim. And then orange is the CPI method, where you don't make any assumptions about who's in there. And again, it's getting the same numbers at around 7 zeros. What's most important about this picture is of that whole list of evidence items, only 30% of them have a human statistic. So the other 70% are not getting any number at all. And that is a situation that was from data from 2010, 2009, that's only gotten worse. So that's what's not being reported. It's not just that the computer is getting a lot more information, it's that it's producing results when people just say nothing. And it's producing results that other methods that the crime labs use discard. This is, again, we're presenting at the subcommittee. It's 2010, it's 2011. This was presented in the spring of 2011 by Joe Goldie, who runs the crime lab in Suffolk County, New York. And this is a study that we had done with him. Here are 52 match statistics from cases that he was interested in. The light blue bars are showing TrueAllele's match statistic results. The red bars are showing what his lab produced as a statistic, using the generally accepted approach of the forensic community. You see 2 things. The computer often gets higher match statistics, but more importantly, the computer is reporting on 27 items that a person's giving as an inconclusive and giving no results at all. So more than half the time, the computer's getting a result and a person is not reporting anything. Okay? So after 2 years of going back to the DNA subcommittee, in May of 2011, the subcommittee provided a binding recommendation to approve TrueAllele for casework in Albany. And that was ratified the next month by the Commission on Forensic Science. Okay. And we had a user meeting this year. 10 groups who are crime labs that are in different stages of bringing TrueAllele on board showed up. California and Virginia are both live and using it routinely in their casework to solve mixtures. We also had prosecutors attend from Schenectady. Whedon Wetmore gave probably the best talk of the meeting. It was just really stirring. It wasn't long. It was just really good talk about a case. There were some prosecutors from Westchester County. We had worked on a case that resulted in a guilty plea of a man who had essentially raped his daughter weekly from age 7 to age 14. It's a long story. It's a terrible crime. And the reason that we were involved is that crime labs feel they can't separate out mixtures of relatives. And so we actually get a lot of parent-child rapes as part of what we see. Okay. TrueAllele has been used in New York to analyze DNA evidence. At this point, only Cybergenetics is doing it. I'll say more about that in a second. These are counties, Cayuga, Chemung, Onondaga, and so on, where we've issued reports. Chemung, there was a trial, the first time TrueAllele went to trial in New York State. That was in September for a serial rapist. Here we talked about Onondaga, the beer bottle, and Westchester, I mentioned what that crime was, usually for murders and rapes. And TrueAllele has been used in criminal cases, for over 200 cases, state, federal, military, and foreign courts, for all sorts of crimes, usually serious crimes. We don't get much by way of larceny, I mean, usually something major. In terms of admissibility, this is in the US. The first time TrueAllele was used in Pennsylvania, there was a Fry challenge. It was admitted by the trial judge and confirmed by the appellate court in 2012. In 2013, there were Fry challenges in California and Virginia, and TrueAllele was admitted in those cases. And this year so far, there were Daubert hearings in Ohio and Louisiana, and TrueAllele's been admitted after those challenges. And we had in Schenectady, New York, a Fry hearing, and that outcome is pending. Most TrueAllele cases, well, they could be an inclusion or exclusions or whatever it is, but if there's a match statistic that includes someone, usually results in a guilty plea. They can go to trial. I've testified about 20 times in trials in the last 3 or 4 years, and every now and then there's an admissibility challenge, and it's a lot more work for people to prepare, but fortunately prosecutors are very excited about learning all of the stuff they never knew they'd ever have to learn about. Okay. In the US, TrueAllele's been used in labs or case reports in about half the states. And I just want to wrap up by talking about what's going on in the country right now. So in Virginia, we saw there were 375 cases where the reports had to be pulled back. And if we extrapolate, we say that's an annual rate of uninterpretable evidence, because that's only very serious crimes. Then you can extrapolate by multiplying by the number of years, the population size, to maybe 60,000 inconclusive cases over the last 4 or 5 years in the US. In New York, we even have a tighter number. Earlier this year, Dr. Barry Duesman, who is director of the Albany Lab, asked his staff to do an inventory one week. He said, okay, just count. How many cases did you get? How many inconclusives did you call where you believe that had you used the TrueAllele system that we're poised to use, based on your experience, you would have gotten your result? They said, well, we had 40 cases, and 20 of those we had inconclusives where TrueAllele should have gotten an answer. That's about half. And so if you multiply by 50 weeks, that's 1,000 cases a year. That's the estimate from the lab. And the lab has had TrueAllele and been allowed to use it for 3.5 years now. So that number of 3,500 is about 10 times in New York what it was in Virginia. And that extrapolation, again, gives a number on the order of 5 zeros, about 100,000. And so it's well known in the US there's a massive underreporting of DNA statistics. Either results are called inconclusive or insufficient, or a lower number is given, or a random number is given, but that's another talk that I gave. And so this is evidence that's been collected and analyzed, and it's been tested. It's been paid for. Right? You have the new initiative coming out of New York City for $35 million to test all these rape kits. I got emails from people in New York State at crime labs saying, okay, and then they're going to throw out half of it? If you understand the issue, it's great to spend all this money on testing, but why isn't it being interpreted? Why aren't you using the data you already have paid for and you have? So this is the situation with the New York State Police. They received approval about 3.5 years ago, and they've done a tremendous amount of excellent stuff. They had their approval. The systems were installed. They have enough capacity to do probably the whole eastern part of the United States. Their systems are up, they're running, and so on. They've done validations. They've not only done the validation studies that we've published, They've done more internal validations when a new director arrived a year ago. They've looked at 3-person mixtures, 4-person mixtures. They've looked at relatedness studies. And the results of those reports, which they're presenting at scientific meetings, are that TrueAllele is very reliable and we need to start using it. That's the scientists' view. We've trained over 30 people. There's a core group that's even super trained, but they're ready to go. They've done their certification. They have their protocols. Everything's written. And a lot of management changes. There's a new director who came in October, the week they were supposed to go live for the umpteenth time. And there's been more and more delay, most recently when prosecutors inquire, when are you going live, for the last few months. Well, we're working on the language of the match statistic, which they have been thinking about now for 3 to 6 months. But that's in the face of 1,000 cases a year that are not making its way as match information for free to prosecutors that could be going out there. So a lot of things are important, but I don't think they've actually heard from many prosecutors asking, well, it would be good to have the information. Maybe it would help us in our case, promote public safety, reduce crime, criminal justice, the sort of things that you care about, because that's your job. It's not the lab's job. The lab's job is to produce excellent data. And some labs, like New York, have gone to a great extent over a period of time to be able to do more with the data. But there's no requirement that they turn on the switch. No one's forcing them to. The accreditation bodies aren't. Washington isn't. There's no social force to ask a crime lab to do more with the data that it has. So you know where they are. And thank you very much. There's more information on our website. There's about 50 to 100 YouTube videos of talks like this, of different presentations. Any publication we're allowed to post, we have it posted. We give webinars of whatever the community is asking for. New York State gave a webinar last year about its validation. I just gave 2 webinars last month and this month for forensic scientists about the details of how the system works to just make it more and more transparent. We publish the map, which you're probably not going to read, but it's all out there. And so thank you very much, and I'd be happy to entertain questions.